Today we're going to lecture on Chapter 7. All the other chapters have been presented in lectures and are available as recordings on the website, as you know. So we are now, I'll switch to the computer here, beginning to discuss cellular neuroendocrinology, Part 2. So we are, in essence, as you'll see, reviewing some material. We're beginning to layer on material as we bring in the concepts that we have put in place previously. And today we're going to focus a little bit more detail on synthesis and continue building our background on regulated gene expression in that context before we get to any specifics in the future. And we'll talk about release, the exocytotic release. And so today my expectation is that we'll be able to complete a discussion of Chapter 7, and then Chapter 8 will be from this series of slides, but posted as a separate recording. So we're going to focus from Chapter 7 on the regulation of the hormone synthesis, storage, release, transport, and deactivation. So we've talked about many of these concepts as we have proceeded in just studying the neuropeptides in general, and that's why I wanted to talk about those first and then talk about neurotransmitters. Then we can begin to compare and contrast on a bit more of a detailed level as we get down to focusing on regulated release as well as regulated synthesis, because we know that we always couple that. When we talk about one hormone, if we use the neuroendocrine system as the model, one hormone, say a hypothalamic releasing hormone, binding to its cognate receptor on its target cell, somehow the response to that binding, the ligation of the cognate receptor, we're going to change the inside of the target cell so that we couple our response, it might be diverging pathways, to stimulate release and synthesis concomitantly in that target cell. We've developed the appreciation for that concept as we have gone along, and still we'll keep them somewhat separated and then progress to getting into the specifics of these divergent pathways, especially when we focus on receptor-mediated mechanisms. So we'll talk about the chemical structures of the hormones a little bit more, the hormone synthesis, storage, and intracellular transport of the peptide hormones, the hormone release, hormone transport, and the deactivation of the hormones. So in this sense, we're also developing our appreciation and understanding for the life cycle of a hormone. Any molecule, biomacromolecule that has a biological activity or function is going to have a life cycle. You have to synthesize it, maybe in an inactive form, a readily available pool that can be activated. It has to perform its function, be inactivated, and either recycled to be activated again through the inactive pool, or be degraded to be eliminated. So this is that general life cycle of a biological macromolecule that we keep in mind. So we've got an understanding of the structure itself related to the synthesis. How does it get to the point of doing what it's supposed to do, and then how do we remove it from the biologically active pool for recycling? We don't want old hormones floating around as well. We need to remove that signal, as we have stated before. So in Table 7.1, we can just read through the different types of hormones relative to their chemical structure. First, there are the non-steroid hormones. So these, as we've seen, are modified amino acids or hormones synthesized from single amino acids. These, for example, can be the catecholamines, so adrenaline and noradrenaline. We've always reminded ourselves that not only do they act in a neurocrine fashion between neurons, but also they're released by the adrenal medulla, and there is spillover from the autonomic nervous system, the sympathetic nervous system, and specifically for these two, so that you can detect these two molecules in the blood, and they can target various tissues and organs and cells, including the anterior pituitary cells, as discussed in Chapter 6. So these are synthesized from adrenaline, And the biosynthetic pathway for the synthesis of the catecholamines and the indolamines is described in detail in Chapter 5. This is taken from Chapter 5. There's a reference to Chapter 5 in this table here, and I have added additional detail in the lecture for Chapter 5 that is recorded and available on the website. The indolamines include melatonin and serotonin, and they're synthesized from tryptophan. So serotonin is synthesized by widely distributed single cells throughout the gastrointestinal system. As well, we know the pineal gland synthesizes and secretes melatonin at night and serotonin during the day, and the biosynthetic pathway for these two molecules is also described in great detail in Chapter 5. I remind you that the structures for these molecules and the biosynthetic enzymes are discussed at varying degrees of detail, and you are expected to know and understand and be able to recognize it when it comes to the structure of these molecules as far as the testing of your working knowledge is concerned. The thyroid hormones, the thyroxine and triiodothyronine, are synthesized from iodinated tyrosine. We'll discuss that in detail when we talk at a more detailed level about the hypothalamo-pituitary thyroid axis. We know very well already that there are many peptide hormones formed from chains of amino acids. So we've progressed from single amino acids that are modified to chains of amino acids. So there are the small peptides, and we talked about thyrotropin-releasing hormone. We'll talk about it in this lecture back to the level of the gene as far as it being a 3-amino acid or a tripeptide, the smallest known yet, other than those that are just modified amino acids. And angiotensin 2 is an octapeptide. We'll talk about it with reference to the prohormone and the enzymatic processing of prohormones. Vasopressin and oxytocin are both nonapeptides, 9 amino acid residues. We talked about those in great detail already using material from the Endotext online in a prior presentation. The LHRH, I've informed you already, whenever you see LHRH, you immediately change that to GnRH. The LHRH is old nomenclature. It is not the accepted term for this decapeptide. Somatostatin has 14 amino acid residues, and as I said, this is where my ability to do the nomenclature, I know the unidecapeptide is 11, and dodecapeptide is 12, and after that, I'll just begin referring to them as amino acid residues and discard that type of terminology. Then we proceed from the level of 14 residues to the larger ones, such as gastrin, 17 and 34 amino acid residues, so two different forms, isoforms of the same hormone. Secretin, 27, glucagon, 29, calcitonin, 32, and ACTH, 39. So we see that we're building on the size of these. And, of course, only one of these is an anterior pituitary hormone. Excuse me. In the prior list, we saw 2 posterior pituitary hormones, a neuropeptide, as well as a peripheral hormone, and 2 hypothalamic releasing, 2 releasing hormones and 1 inhibiting hormone, the somatostatin. So there's no clear distinction. The point there being made that small ones are not reserved for hypothalamic releasing hormones, and then they get bigger in the anterior pituitary, and then biggest in all the peripheral organs. It really is intermingled throughout. It's specific for each peptide. And then some of the peptides, there are many different forms, like CCK is a prime example. There's a CCK5 and a CCK8, for example. So there can be many different biologically active cleaved molecules from the going way back to the original pre-pro hormone. So again, it's the specifics. But you should familiarize yourself with these, at least being able to categorize them on modified amino acids, small peptides, the large peptides, and then the proteins are actually polypeptides, because we're not talking about hundreds of amino acids, acid residues like some of the enzymes. We're just talking about 51, 84, 191 for human growth hormone. So this is an anterior pituitary hormone, really one of the larger ones that are present. To come back, we see that some would be on the edge. I know that corticotropin releasing hormone is 41 amino acid residues. So is that a large peptide, or is that a polypeptide? So we won't get too concerned about the distinction for the ones on the border, such as that. There are also some of the hormones that are glycoproteins. So there's hormones synthesized with the post-translational processing, such that a carbohydrate, typically a chain of carbohydrates, is added to the protein. For example, FSH, LH, thyroid stimulating hormone TSH, and human chorionic gonadotropin. You note that 3 of these are gonadotropins, the FSH and LH, and the HCG, which of course is the pregnancy hormone that is produced by the primordial placental cells, if you will allow me to use that term, and the placenta to replace the pituitary gonadotropins during pregnancy, and we'll discuss that in great detail later. These 3, I know, I'm not certain of the chemical makeup of this, but these 3 are heterodimers. They share one subunit, alpha subunit, in common, but their beta subunits are actually separate, and we'll get into the details of that as we proceed through the semester. So what this reminds us of when we talk about hormone synthesis, if they are dimeric, and especially heterodimeric, and in this example where there's a sharing of one of the subunits, the alpha subunit, then we would start thinking about tissue-specific gene expression and cell-specific gene expression. The gonadotropins and the gonadotropes, and the thyrotropes both express the gene for the alpha subunit, exact same proteins, but they express the different beta subunit genes, the FSH and LH and the gonadotropes, and the TSH beta subunit for thyroid stimulating hormone. It also reminds us that there's other post-translational processing that is occurring for these large structural associations between the subunits, or the assembly of the subunits. So in this one list, we have 2 examples of post-translational processing, other than thinking back to the cleavage from the pre-pro hormone to the pro hormone, and the folding such that the subunits are ready to be assembled. One of the ones illustrated just in this list. And then, of course, the other is the glycosylation, the addition of carbohydrate side chains. Then the prostaglandins are synthesized from essential fatty acids. They are important in the immune system, but more importantly, more relevant for our course in the menstrual cycle. We'll spend quite a bit of time on the steroid hormone synthesis later, but at this point we want to emphasize the 3 general categories of gonadal steroids. They are all synthesized from a common precursor, cholesterol. The cholesterol is stored as a cholesterol ester, so it has an ester bond that links it into the tissue. If you didn't esterify the cholesterol, it would be lipid soluble and freely diffusible. So that's the only way to store up any steroid hormone, if you want to think of it as a storage mechanism. Once you begin synthesizing these, it goes through a biosynthetic pathway, one enzyme to another. The intermediaries are typically converted to the next molecule in the stage, so it doesn't accumulate enough to diffuse away. It's enzymatically converted. But as soon as we get to the final, or any point along the biosynthetic pathways, where there's any level of accumulation, some of it can escape by diffusion. So as soon as the gonadal steroids are produced, they are released, they diffuse away. Okay? So there are three categories: the progestins that include progesterone, which is the major one that we know of and is the predominant progestin in most animals and in humans. So we typically just refer to progesterone. The androgens, testosterone is the major one that we think of, and we'll identify others as we proceed and look at the enzymatic pathways for the conversions. Of course, testosterone can be converted to estrogen, the 17-beta estradiol, the major estrogen that we call estrogen or just estradiol, but there are many other forms of estrogen. Most of us know, especially if we're all stuffed up and inflamed as I am, and if it's with allergies or for other type of inflammatory symptoms, we are given prednisone, and prednisone is a common precursor along the biosynthetic pathway for these, but it more selectively elevates the levels of glucocorticoids, especially cortisol in humans, and brings about that anti-inflammatory response. You also, if you've ever taken the prednisone or any other type of steroid as an anti-inflammatory treatment, you know that you take higher doses to get started, but then you have to take smaller doses for a couple days to taper off, and we think immediately, why would we have to do that? And of course, negative feedback is the answer. If you add exogenous precursors even that build up the cortisol levels, and then suddenly, and then that begins having the negative feedback at the CRH and the anterior pituitary, then you need to taper exogenous and let the endogenous levels come back up and balance out the whole system. Otherwise, you will enter into an immediate state of hypocortisolemia until gradually the system will readjust and compensate and get back to homeostatic baseline levels. So we can keep adding in these things, and what's important is that you develop the skills as well. As soon as you see glucocorticoids, corticosterone, that's for non-humans, especially rodents, this is the major form, or cortisol, all these other pieces of information stuck away in your brain somewhere come flooding to your forebrain, your prefrontal cortex, and you can put it all together and have a clear picture of a concept and be able to think about it critically just from one word on a list triggering these neuronal circuits. Don't get too stressed out, because your cortisol will inhibit your cognitive function, as we know, and maybe kill some of your hippocampal neurons, so don't get too stressed out about it. And then the mineralocorticoids, of course, includes aldosterone, which is the primary one, and we'll spend very little time talking about the function of aldosterone in this course. I know this is a difficult slide to read, but I want to read through it because it is a nice overview of transcription and translation, and it should all make sense to you, and I want to continue building it into one big picture before we jump into any specifics and start focusing in very closely on transcriptional regulation and promoter regions and things like that. So we start first, of course, as we know, with the DNA, transcribing to RNA by the enzyme RNA polymerase. I think on one slide I had that transcriptase, and it was, of course, wrong. It's a polymerase. It's controlled by promoters, so we're going to spend quite a bit of time talking about promoter regions. Controlled by regulatory proteins, the transcription factors. We've already talked about some, and we'll build on that. We'll give some examples in this lecture and an upcoming slide, actually. Controlled by cell state, that is, if the cell is dividing. Of course, neurons are postmitotic. By far, virtually all of them. There's just a very small population of neuronal precursor cells that are capable of undergoing mitosis. The functional neurons are all postmitotic. I'm actually not certain if there are precursor tropes in the anterior pituitary, to be very honest. But in most glandular tissue, there are some reserve cells that can go on to differentiate. RNA transcripts are subjected to post-transcriptional modification, the RNA processing, as you know, and control. So ribosomal RNA transcripts cut into the appropriate size classes for the initial assembly in the nucleolar organizer region. Transfer RNA transcripts have to fold into shape. You know, they're the typical hairpin type of shape as they're described. The messenger RNA transcripts get the 5' cap, as we know, poly A tail, the non-coding sequences. The introns are removed from the interior of the transcript. There's a little more information on this splicing that we'll talk about in an upcoming slide. All the RNA types must move to the cytoplasm via the nuclear membrane pores, so we remember that. The mRNA molecules are translated by ribosomes, the RNA plus the ribosomal proteins, which match the 3 base codons of the messenger RNA to the 3 base anticodons of the appropriate transfer RNA molecules. Actually, a few people forgot that the transfer RNA is the RNA molecule that brings the the amino acid in to be added to the growing polypeptide chain. So reviews never hurt. The ribosomes initiate using the first AUG codon to start protein synthesis, with the first amino acid being methionine. The message is read 3 bases at a time. We know it's a triplet. Consecutive codons, so each codon is a triplet. There's no commas or overlap, so it should have a high fidelity of reading 3 nucleic acids, 3 bases at a time. The peptide bonds are made between the adjacent amino acids to produce the growing polypeptide chain. And then when a stop codon, the UAA, the UGA, or UAG is encountered, then the translation ceases. So how do we know this is messenger RNA, or RNA here? Because of the U. That is only present in the RNA. So the newly synthesized proteins, and we remember that translation can occur at 2 places: the free ribosomes for what we call the general housekeeping enzymes. From Chapter 5, we are reminded that some of the biosynthetic enzymes for the monoamines are also found free in the cytoplasm. Therefore, they are translated at the free ribosomes. So it's not always just these general housekeeping proteins, but typically that is the collective phrase, as we have stated before. There is going to be some modification, the post-translational modification. Proteins undergo a final conformation adaptation in conjunction with the chaperone proteins. So the folding doesn't occur all alone. There are chaperone proteins that help with that. The soluble proteins may have the sugars added. Secreted proteins must be synthesized through the membrane, and the initial portion of the protein is removed in the process. The signal sequence of the nascent peptide, as it is inserted through the ER membrane into the lumen or the cistern of the ER, rough ER, of course. Multimeric proteins must assemble from subunit proteins. We just used the gonadotropins and TSH as an example of that. Some proteins, such as insulin, have a portion of the protein removed and discarded, so the enzymatic cleavage we know about. And then the protein carries out its function. So this is from gene to functional protein in general, the generic approach, but it's a nice way to just stop and think about it. And we have many examples that should be flashing through our mind already, and we'll look at a few more. Right at the present, though, we should be stating the obvious to ourselves. There's a number 6 on here, and this is removal or metabolism, specifically catabolism. As you know, metabolism is anabolism, the making of more complex molecules from less complex molecules. So everything up to this point might be considered anabolism. And then catabolism is breaking down functional larger molecules to non-functional, typically non-functional smaller molecules, or less biologically or inert as far as biological activity is concerned. Well, now a few comments on the controls over gene expression in eukaryotes. We've talked about it before. We're always thinking of cell-specific gene expression, tissue-specific gene expression, maybe even organ-specific gene expression, and definitely different genes among different genus and species and strains of animals. And there's even allele differences, but let's stay away from all of that. So the genes may be deactivated by histone or nucleosomes. So that's tighter and tighter coiling to generate the heterochromatin that we talked about way in the first lecture. Heterochromatin has a lot of these histone and nucleosome proteins associated with it for the coiling and supercoiling and all that stuff. So definitely not available for gene transcription. But even in the euchromatin, there's different degrees of coiling of the DNA, and even to the level of the alpha helix. So if we can relax that alpha helix, we can make the promoter region and the gene more available to the proteins that have to come in and act on it, the transcription factors and the polymerase. Methylation of the nucleotides can also deactivate genes, and coiling or condensing of the chromatin as we just described. There's also transcriptional control once you have transcription established for a cell. It includes reversible binding by hormones. We'll definitely talk a lot about those. We'll see some examples of heat shock proteins and also other examples of transcription factors, inducers and repressors that we won't focus on a great deal in this class, but there's plenty of other classes that you can take to busy your mind with those ideas. This information I have provided the source in the upper part of the slide, and we're thinking of the transcription factors, the list on the left, the sequence motif in the middle column, and the comments to describe these. So let's think about some of these. The C-mic and the MAX are transcription factors that we won't deal with much. But it's first identified as a retroviral oncogene, and the MAX specifically associates with the C-mic in cells. So these two form a heterodimer to become functional and bind to this CACGTG sequence motif. So how do we know this is DNA? Because we have the T and not the U, right? The CFOS and CJUN, these will, we have mentioned these. These will be discussed in greater detail. We talk about regulation of gene expression, especially in the neuroendocrine system. They become phosphorylated and form heterodimers, and the sequence motif is the TGAC, GTCAA. So, the challenge for you, of course, is to try to recognize these. I won't overdo this type of thing. You'll find plenty of classes where you'd be expected to memorize entire genomes, so this is asking just a little bit. But what we want to do is compare it to the CRE, the cyclic AMP response element, to which binds the transcription factor CREB, the cyclic AMP response element binding protein, which when phosphorylated binds to this. At first, let's note that the CFOS, CJUN, both first identified as retroviral oncogenes. So I told you that this is feline osteosarcoma. I'm not sure of the name of this one and why the letter C, but anyway, that one sticks with me and reminds me of the retroviral oncogenes associated in many cells throughout the body, and it is also known as the AP1 factor. So this is called the AP1 consensus sequence, or response element, regulatory element was the other term that we used for this. So that's the AP1. We'll hear about that. And this is the CRE. We note that it's T, G, A, C, and GC, and TC, AGA. So there are some differences between these two, but not dramatic differences. These also are short sequences for these proteins to bind to. So these are, in my mind, some of the impressive aspects of the sensitivity of DNA strands to transcription factors. Such a short sequence can bind a protein and dramatically impact gene expression. We'll see that the CRE and the CREB and cyclic AMP and the adenylate cyclase coupling to the receptors for the hypothalamic hormones is the key link between hypothalamic releasing hormones and the divergent pathway coupling secretion with synthesis at the level of the gene. So the CRE binds to the cyclic AMP response, the CREB, CRE binding protein, binds to the cyclic AMP response element, the CRE. This is a family of at least 10 factors resulting from different genes or alternative splicing. So remember, we talked about the splicing out of the introns and keeping all the exons. Well, there's alternative splicing where you definitely get rid of the introns, and sometimes you get rid of exons, and you have actually from an identical single gene, you can produce two isoforms of the protein. One would have an exon included in its amino acid sequence ultimately, and the other one would not. But they're still in the same family, and can still have similar biological activities. But some different regulation would be suspected if you have isoforms. Different isoforms are expected to behave differently under different biological conditions. Anyway, the CREB can form heterodimers with the Jun. And typically for I know at least for these transcription factors that we will be dealing with, they they have a low affinity for their consensus sequence, their regulatory element, in other words, in the unphosphorylated monomeric form, and a very high affinity in the phosphorylated dimeric forms. So therefore, kinases are going to be very important in targeting these substrates to phosphorylate them, such that now they have a higher affinity for each other, and after forming the homo or heterodimers, have a high affinity collectively for their regulatory element, their cognate regulatory element on the promoter region. The mRNA processing, as we just said, reminded ourselves, includes the intron removal or that alternative splicing, and we'll use some examples for this. In the Chapter 5 lecture, we talked about a synthetic enzyme for catecholamines, the tyrosine hydroxylase, that converts tyrosine to L-DOPA. In humans, in humans and in rodents, there are at least two isoforms of tyrosine hydroxylase. However, when the search ensued to the point of finding and isolating and characterizing and sequencing the genes, there are two different genes for tyrosine hydroxylase in rodents and only one in humans. So from that we can deduce that in humans, to get two isoforms of the enzyme, we must have alternative splicing so that one gene produces two distinct but functionally similar, of course, gene products. So that's the example in humans of the tyrosine hydroxylase gene generating two different proteins. And we saw that CREB is also another example of alternative splicing, and we'll build on that list as we proceed to the semester. The degradation of the messenger RNA, so any biological macromolecule has a half-life. It has a life cycle. So messenger RNA is synthesized, it is altered so that it becomes biologically active, and it is biologically active or available to be biologically active, and it is capable of being degraded. So there is a pool of messenger RNA that might be sitting there in the cytoplasm, but not necessarily being translated. It's available for the degradation mechanisms, and if it doesn't get used before it's degraded, it's lost. Definitely briefly after translation, the messenger RNA is highly susceptible to degradation and will be degraded I don't know exactly what the time frame is, and I don't know if anybody knows the exact time frame, but that's when it is most susceptible. I think in a subsequent slide there's description of the mechanisms for protecting the mature messenger RNA. Actually, this is one here, but it also can bind to other proteins and just be sitting in the cytoplasm, safe from degradation. The nucleases, RNases, any of you that work with isolating messenger RNA know that RNases are just about everywhere, so there's plenty of opportunity for RNases to degrade messenger RNA. So there's proteins that can bind to them just to protect them from the RNases, and the length of the poly-A tail also, as it increases in length, the stability of the messenger RNA increases. How this is regulated, we don't know 100% yet, but it can differ from time to time. So there's still so much that we don't know, but it is always very interesting to expand our understanding of this. Translational controls, we store the mRNA in the cytoplasm until needed. So this is what I was saying. There are protective proteins, riboprotein complexes sitting in the cytoplasm. It's very important to understand this concept because one of the methods that we'll be talking about, or if any of you out there taking this course are involved in research or end up in research where you are measuring messenger RNA levels by whatever method, must understand that if you have a certain amount of messenger RNA, a number of copies of messenger RNA per cell, it doesn't mean that that is directly correlated with translation and the amount of protein. And of course, the amount of protein isn't always correlated with the level of biological activity, because we have the inactive pool and the active pool of protein. We have the inactive pool and the active pool of the messenger RNA. So if you just measure the total amount of messenger RNA, it's a safe index of protein synthesis, and we can go back to gene expression even and take a look at that. So we get these static pictures sometimes, and we have to be very careful how we interpret the data. Higher levels of messenger RNA are a safe and reliable index of increased translation, but you must measure the amount of gene product as protein, and without a functional assay of the protein, you don't know if you really are measuring an index of altered function in that cell. So these details become very important when you start looking at research reports where people measure the level of a protein and then go on to conclude that they've indicated a change in the activity of the cell, and if they don't get some index of the function of the protein or rate of translation, which is rarely done, they cannot draw those types of conclusions because their evidence doesn't support that conclusion. So here the stability of the RNA, the riboprotein, the RNA complexes in the cytoplasm. So again, emphasizing the life cycle of a molecule. Synthesis, storage in sense, a pool that can be activated, so an inactive pool to become activated, or a readily releasable pool for hormones is what we'd be thinking about. And then the biologically active through activation, biological activity, and then termination and destruction of biological activity and biological structure. So once they're produced, they're controlled. The signal sequence is taken off. It's cleaved almost immediately in the ER. It can be stored and processed in the ER, as we know. Glycosylation and cleavage can occur here. Storage and modification in the Golgi apparatus. Can be glycosylation there. Association with other cleavage enzymes. Typically, there isn't much cleavage that occurs in the Golgi apparatus. It'll usually be in the vesicles in which the Golgi apparatus has assembled the cleavage enzymes with the appropriate proteins. And then vulnerability to degradation within the cytoplasm is also a consideration at the level of the protein, of course. So many different levels of detail and control, and we'll build on them as we take it case by case and discover maybe a research article that gives us more and more insight. So what are some of the advantages of prohormones? Well, I think already with the discussion of the life cycle of a molecule, it's quite obvious what we need to do. This enables the three-dimensional structure of the peptide that would be stabilized during the synthesis. So you get a little extra bulk to the molecule so you can twist and turn and then perform the cleavages and glycosylations along the way. We saw examples of that with the oxytocin and vasopressin and the neurophysin carrier proteins for those, and C-terminal, I think, peptide on one of them that became glycosylated. Who knows why? It doesn't become a biologically active portion, but that might have been for stabilizing the molecules to produce the three-dimensional conformation. And as you recall, there were the two cysteine residues that get involved in a disulfide bond linkage in the mature functional hormones. Prohormones are easier to transport and package into granules than active hormones. That makes sense. I don't really know why or how, but that's the claim that we see here. It may just be that you have bigger molecules. Well, for example, we saw the neurophysins still have the hormones, vasopressin and oxytocin, attached to them and form a homodimer, and that's what gets shipped to the Golgi and placed into the vesicles, and the final cleavage of the neurophysins from the now biologically active hormones takes place in those vesicles or secretory granules, the term we should start using now, so that we have the active hormones at the site of release at the neurohemal junctions for the neurohormones as we're considering for the moment. They can act as storage reserves. So you've got this pool of secretory granules. If you recall from test 1, there was the illustration of the neurohemal junction in the posterior pituitary, where the axon terminal had a big bump in it, and this is where the stored pool of secretory granules was located, and then the actual terminal. And I'll be damned, I can't remember what those dilated pre-terminal axons in the neurohypophysis are called, where the hormone is stored. But you would expect that between that storage reserve site and the active grid, the presynaptic grid, where you're going to have the exocytotic release, that between the movement from one location to the other, you would definitely have the final cleavage of the prohormone to the biologically active hormone, so that the full biologically active oxytocin or vasopressin would be released into the blood. So it can be used to regulate the amount of hormone released. I don't know how that's carried out. I'll just take the author's word for it. Are more resistant to degradation than our active hormones and have longer half-lives. So we've already talked about other modifications of some of the hormones, and we'll continue to bring those up when we talk about it case by case, that extend the half-life by making the molecules less sensitive to degradation. We've seen a few examples already, and we'll build on those as we proceed. So the final thing that I'll discuss in this lecture is the TRH gene. So this is a schematic illustration of the rat pre-pro TRH gene, and the source is shown here. The genomic DNA is at the bottom, and the messenger RNA is at the top. We notice that this is 5' messenger RNA to 3', so this would be 3' to 5', right? On the DNA, the start site would be somewhere here, so the promoter would be upstream of that. As we go downstream, we encounter exon 1, intron 1, exon 2, intron 2, and exon 3. And then the termination site would be located here. So exon 1 produces a new concept that we will see repeatedly, the 5' untranslated region of the messenger RNA. So it has other purposes. The first part of exon 2 produces the putative signal peptide, targeting it for the rough ER. The signal peptide sequence, it must be present in the DNA and the messenger RNA for the ribosomes to bind to it and bring it to the ER to produce, to insert the developing, trying to get my pip set here, the growing peptide, that's the term I was trying to think of, into the rough ER. So, and this is the signal, this produces the signal peptide that is cleaved almost immediately after the peptide is growing inside the ER. So the first cleavage takes place very early on. So exon 2 contributes to this part, exon 3 to the remainder, and the 3' untranslated region. So this is introducing to you, notice the scale differences. This is 0.1 kilobase pairs and 0.2 kilobase pairs for the DNA. This introduces to you that we will talk about all the genes, how many exons, how many introns, which exon contributes to which part of the messenger RNA or to the peptide itself. And there's always going to be a 5' untranslated region and a 3' untranslated region. And then the final point I will make about the TRH, actually I'll take just a few extra minutes to go to the next couple slides as well. But the first point is that there are 5 glycine, histidine, proline, glutamine, histidine, proline, glycine sequences in the pro-TRH hormone, and you see them here, 1, 2, 3, 4, 5, they are underlined. So one TRH gene can produce 5 TRH molecules. So the post-translational cleavage of the pre-pro TRH has to be extensive, and then these end up being tripeptides, as we know, and this is the molecule of the TRH. We'll go through this again when we talk about the HPT axis specifically. We're using it now as an example of post-translational processing of hormones, especially the hypothalamic hormones in this context. The next couple slides show the use of antibodies specifically to a portion of the pro-TRH, one of these other parts that is not any one of these portions here, so anything that isn't underlined could have been what was targeted to produce this antibody. So it's specific for the prohormone, the pro-TRH, and this is a cross-section of the rat brain, the third ventricle here, so this is the para and periventricular nuclei, and we see TRH prohormone containing neurons, you see them here, but there are no axons emanating from those neurons, and here in the median eminence, no axons detected whatsoever. The axons are there, but the pro-TRH is not detected in them. So this means that if we could go into these cells and look at them with the electron microscope, we'd find the pro-TRH stain in the ER, in the Golgi, and in the transport vesicles just close to in a peri-Golgi region. And the farther away from the Golgi apparatus those vesicles get, the less of the pro-TRH staining we would be able to detect. Now, the peptide after cleavage is still there, but it doesn't bind our antibody anymore. Now, if we use an antibody that stains for TRH specifically, and we look at the median eminence, it still stains the neurons because the TRH is still within the pro-TRH, but the TRH now stains the median eminence. You see these terminals all around the primary capillary loops in the external zone of the median eminence, here in the stalk median eminence, the pituitary stalk, and even in the pars nervosa, as it turns out. So I'll end with that slide, and we'll introduce the angiotensin II in the next part of this and continue on this discussion to complete Chapter 7 and go on to Chapter 8 in the next recorded lecture. I appreciate your time and attention, and we'll take a moment here if I can grab the camera and zoom in on my get it straight here. Endocrinology meeting 2002 in San Francisco. I didn't get to go, but a student did go and came back with a shirt. All I got was this thinking shirt. But it reminds us that there are societies in endocrinology and neuroendocrinology, national, international, and there's a tremendous breadth of research, even behavioral neuroendocrinology societies. And there's a tremendous number of scientists investigating neuroendocrinology day after day, and an endless number of research articles that we could use for the course, but we'll begin to introduce some from here on out. So see you all again soon. Bye-bye.